Hi guys, welcome back to my channel. Today we're going to be talking about carbohydrates. Usually in all of your biology focused classes, we're always going to start with the 4 biological molecules. And today we're just going to go over carbohydrates. So what do we know about carbs? So essentially we know that carbs have a 1-to-1 ratio of carbon, hydrogen, and oxygen. All carbohydrates will have this. So it'll look something like this. Chew, whatever you want to call it. I know chew is a way to remember it if you want. So it'll look something like this, or you might see it like this as well. So a hint to know that it is a carbohydrate, or a hint to know that it is a sugar, is if it ends in ose, as I mentioned right here. Carbs are sugars, y'all. So anything that ends in ose is a sugar. Anything that ends in ose is a carb, because sugars and carbs are the same exact thing. We actually get most of our energy from carbs, so don't cut those out of your diet, even if everyone is telling you to, because you will feel like poop, I promise. Now, we're going to go into monosaccharides, which is our first little subclass of carbohydrates. Mono, as everyone knows, means one, and if you don't know, that's perfectly fine. Means one. And we were talking about sugars earlier, so monosaccharide means one sugar. And I have this little drawing here for you to show you kind of like the structure of it. Of course, it doesn't have all like the carbons and all that other fun jazz and whatnot, but we don't need to worry about that. I just want to show you the structure of it and how it is the building block. Building block. You might hear monosaccharides referred to as building blocks because they are essentially the base for the longer monosaccharides, if that makes sense. So we're going to get into disaccharides, we're going to get oligosaccharides and polysaccharides, which are basically just multiple monosaccharides. So these are the smallest group of carbs. Since they are the smallest, they can pass easily through the bloodstream, pass easily through the cell membrane because they are so tiny, and also they are polar, so we'll get into that later on when we talk about hydroxyl groups. But you may hear this a lot in every science class that you take. Structure equals function. So I have some people, I work at the training center, and there's this one girl, and I love her. She's just so cool, and I swear this is her motto. Even though I've heard it before, she just like makes it awesome. Like, I don't know, just like hearing her say it makes me like have a good day. But essentially, structure equals function, meaning if, say we have a red blood cell, we know that red blood cells are kind of shaped like this, right? And we kind of have a problem when, say, they're shaped like this. So this is a normal red blood cell here, right? Maybe I should have drew it in red so you know it's a red blood cell, but it's fine. So an original red blood cell is going to have a nice little healthy shape to it, while an unhealthy red blood cell is going to have, it can have a different shape. When our red blood cells are shaped like this, we can have some problems, such as blood clotting, because if we have multiple red blood cells shaped like this, they're just going to start like combining together, and eventually it's going to block the blood, or like the passage of things getting through. And this causes blood clotting, essentially. Alright, so glucose is actually used for brain fuel. Now, what that means is that our brain uses glucose as energy, which, it's kind of important to ingest glucose. So I have two different versions of glucose here. Now, one of them is D-shaped and one of them is L-shaped. So if we look here on the fourth carbon, if we ignore this one, okay, we count 1, 2, 3, 4. We can see that we have an OH bond. So essentially what this is a hydroxyl group. And what a hydroxyl group is, we'll go into that on the next slide. So you may be wondering, what is D-shaped? What is L-shaped? I've never heard of that. So D-shaped just means that this hydroxyl group is on the right side. L-shaped, which this is pretty easy to remember, and as you guessed it, is on the left side. So we have an oxygen and a hydrogen on the left instead of the right, making this an L-shaped glucose. Now, which is better, D or L? In my opinion, I think D-shaped is better, and I'll tell you why. L-shaped is actually, when we are talking about L-shaped glucose, we're talking about things like sugar-free vanilla, Splenda, and all that other stuff. And you may be wondering, well, Sumi, isn't that stuff good? There's no calories in it. Well, that's what they want you to think. But Splenda and sugar-free syrups, such as like sugar-free vanilla, sugar-free caramel, all those other, stuff, we actually can't digest L-shaped glucose. We don't have any enzymes that can digest it. So if we can't digest it, how is it leaving our body? Well, it leaves our body via means of excretion. But if we're having high amounts of L-shaped glucose or, L sugars per se, then we're not, breaking down those L sugars fast enough. We're not getting rid of them fast enough since they don't really have anywhere to go. So most should stick with D-shaped glucose, D-shaped sugars or D sugars, because they're just better for you. Now, L sugars act like D sugars. D sugars are actual glucose that the brain uses. L sugars are kind of like impersonators of D sugars. So when we ingest L sugars, our brain is like, oh yeah, I'm getting glucose. Well, let me put it this way. It's like, well, oh yeah, I'm getting D sugars. So that means I'll have energy, I'll have fuel, blah, 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 blah. But L sugars kind of trick the brain because the brain's like, okay, well, where's the sugar that I was told I was getting, right? And it doesn't get it. So essentially, since we don't have enzymes that break down L sugars, that's not really a good thing that we're ingesting stuff like Splenda and sugar-free vanilla, even though it may seem like a good thing since there's no calories. If we have no calories, there's no energy. So just be mindful about that. I mean, if you want to do that, you still can, but there definitely are some disadvantages in the long run to only ingesting things like sugar-free syrups and Splenda. Now, as I was talking about in the last slide about hydroxyl groups actually make carbs soluble in water. Hydroxyl groups are just these OH groups that I mentioned. Now, why is that? So essentially when we have hydroxyl groups, those hydroxyl groups are able to make hydrogen bonds with water, which is a good thing. So when you have a glass of water and you have sugar, we all know that the sugar dissolves in water. So essentially the molecules from the sugar and the molecules of water pass through the bloodstream easily, allowing for the mixture of glucose molecules and blood molecules so that everything is able to run smoothly. If we didn't have these hydroxyl groups, a lot of our molecules would be repelling each other, which is something that we don't want, because how are we going to get the nutrients we need if it won't mix, if that makes sense. Now there are 3 types of monosaccharides here. I have D-glucose, D-galactose, and D-fructose, and this is just emphasizing the D sugars that I was talking about, or the D-shaped, and just showing you that, you know, we have it here, the hydroxyl groups on all of these, and the reason that they are D is because they're hydroxyl groups on the 4th carbon, 1, 2, 3, or the 5th carbon, but let's just ignore this, let's pretend it's just 4, are all OH, and they are on the right side. So this is just reiterating that. So now I have, this is glucose. Don't get scared. I know it looks scary. I know what you're thinking. That didn't look like the thing from the last slide, and I know. But glucose can also look like this. I just wanted to explain the difference of beta and alpha. So essentially, when you have an OH group on top, so a hydroxyl group on top and a hydrogen on the bottom, the hydrogen on the bottom means beta. So bottom beta, you can think of it like that. Now, you may be thinking, well, this side is switched, so why isn't it alpha? Well, we're starting, if you count the carbons, it's going to go 1, 2, 3, 4, 5, and then 6. And here's that chew that I was talking about earlier in the slides about the 1-to-1 ratio, or 1-to-1 ratio that's in all monosaccharides or all carbohydrates. But anyways, that's besides the point. Since we're starting on this first carbon, we're going to look at this first carbon to determine whether it's beta or alpha. So now if these were switched on this first carbon and the hydrogen was up here and the hydroxyl group was on the bottom, it would be alpha. But since the hydroxyl group is on the top and the hydrogen is on the bottom, for this first carbon, it's going to be beta. Disaccharides. As I mentioned in previous slides, we have di means 2, so we're going to have 2 sugars or 2 monosaccharides. Oh, I'm sorry, I spelled monosaccharides wrong. I just noticed that. But a monosaccharide is 1 sugar, so we have 2 of the 1 sugar. So you can see the structure of a disaccharide is going to look something like this. And if we went back to monosaccharide, it was just going to be just 1 of these, right? So examples of disaccharide include lactose, maltose, and sucrose, as shown on this slide. So if we look at this, we have glucose and fructose that makes sucrose, which is basically just our pure sugar, sugar cane, and we have galactose and glucose that make lactose, which is found in milk, and we have glucose and glucose, which is found in maltose, which is also found in like malt shakes or these little Whopper candy things. Me personally, I don't think I'm a big fan, but you know, if you like it, you like it. It's definitely an acquired taste. But you can definitely see those monosaccharides binded together to create disaccharides. So we have 2, right? 2 monosaccharides making our disaccharides here. So back here, this little image here is just to show you the structure, like the outline of what it looks like. Now, going into monosaccharides and disaccharides, we have specific taste receptors on our tongue that detect whether we like something or we don't like something. So if you bite into like a Granny Smith apple. Me personally, those are like super sour to me, and I don't really like them. And my taste receptors, they pick up on that. Now, my taste buds personally enjoy chocolate ice cream as one of my favorite monosaccharides and disaccharides, because it's just so good. And for me, I taste sweet. Now, everyone's taste pores are a little different, but usually when you eat ice cream, you taste sweetness. And that kind of goes back to kind of what our ancestors did when they were trying out different foods and stuff like that to see what was deemed safe and what was deemed not safe. When we ingest something or like taste something that just doesn't seem right in our tongue, like, or that's just nasty, we automatically spit it out. Like, that's just like a response that we have because it's not satisfying to our taste buds. So our tongue is almost like our defense mechanism against certain things going into our mouths, because if it's not supposed to be in your mouth, your brain knows it's not supposed to be in your mouth. So those receptors are being sent up to the brain and telling the brain this is good or this is not good. So I just thought that was kind of cool to think about. So we are on to oligosaccharides, and this one's going to mean a few. So a few being 3 to 20 sugars, or 3 to 20 monosaccharides bounded together. So essentially I'm going to draw what it looks like on a cell membrane, because oligosaccharides are actually found on the cell membrane, which not a lot of people know that. I was like, why are there sugars attached to the cell membrane? Well, I'm so glad you asked. These oligosaccharides are used as immune defense mechanisms. So they aid in immunity. They also are receptors as well. So they kind of have like a Y shape to them. So this blue thing that I drew is an oligosaccharide. And like I said, those will aid in immunity and kind of cell signaling or receptors, which is kind of cool to think about. So we have 4 different monosaccharides attached together to create stachys. I think that's how you stachys. Yeah, I think I said that right. So that's kind of cool to think about, because 2 of the same 2 monosaccharides create sucrose. 3 monosaccharides create raffinose, and 4 monosaccharides create. So it's just cool to see how they each create different things based on how many monosaccharides there are. Now, this is truly fascinating. So, oligosaccharides are actually found in breast milk of mothers. And what oligosaccharides do is essentially babies aren't born with the most elite microbiome, meaning that they are very prone to infection, illness, disease, and all of the other bad things, right? And we don't want our baby to have that. So mothers actually have built into them their breast milk oligosaccharides that the infant will, will ingest but not digest, because babies cannot digest oligosaccharides or like metabolicize oligosaccharides, so they kind of just sit in their gut. And as I was mentioning before about how oligosaccharides aid in immunity, they actually help the infant with immunity. So they are less prone to disease, they're less prone to illness, they're less prone to other outside factors that could, affect the health of them, because they're kind of building their own little microbiome using their mom's oligosaccharides that are just resting in their little microbiome. So that's kind of cool. Last but not least, we have polysaccharides. Now, polysaccharides are known for their structural support, and they're usually about 20 or more monosaccharides. I spelled monosaccharides wrong, I'm sorry. 20 more monosaccharides bonded together. That's a lot, because there could be just like infinite amounts. But you can find them in plants, like the cellulose of plants, and chitin in animals. So this shell here is actually made of chitin, this lobster shell claw, even though this lobster looks like he's ready to be eaten for dinner. He looks a little too red. And then cellulose in plants. So if you look here, we can see the continuous amount of monosaccharides. We have one, two, three, four, and then it just goes on. It goes on, the list goes on. And it creates these fibers here, which is really cool. So, some examples of this being amylopectin and glycogen. And you may be wondering, what's the difference? Or, aren't they the same thing? If you know what these are, you might be like, aren't they the same thing? Well, essentially, they are. So, amylopectin is actually only plant-based. It can only be produced in plants, as glycogen is only produced in animals, which is kind of cool. Like, they're the same exact thing, but one of them is just produced in plants, and one of them is just produced in animals. Now, if we take a look here at look at the chain. Look how long that is. Those are all monosaccharides bonded together. So a long chain. This is polysaccharide right here, y'all. That's a long chain. Like, look! Oh my god. So, essentially, the amylopectin and glycogen are just long chains of glucose. Which, amylopectin made in plants, just a long chain of glucose. Glycogen, a long chain of glucose, but only made in animals. So, essentially, that is all for today's video. Thank you so much for watching.